In this tutorial we'll focus on mitochondrial DNA. There are several slides of information and this could provide an alternative to your textbook, and most of it's very straightforward and fairly easy to follow, and I think it's very useful. I won't spend a lot of time on each slide. I'll move through it fairly quickly and leave you to pause the video and to read the information in more detail. But I'll just pick up each of the key points fairly quickly. Mitochondrial DNA has become one of the most powerful tools in genetic and particularly evolutionary studies for a number of reasons. So let's have a quick look at what all the excitement's about. The first interesting thing is that mitochondrial DNA is inherited only from our mother, not from our father, and this has to do with the nature of the sperm and the egg meeting in fertilization. Because the sperm contributes very little cytoplasm to the zygote, the fertilized egg, and mitochondria are found in the cytoplasm, this means that the sperm actually contribute no mitochondria to the fertilized egg or the zygote. The egg, on the other hand, we get most of our cytoplasm in our first fertilized egg from the egg itself, and since that's where mitochondria is found, then our mitochondria in all of our cells comes only from our maternal side, our mother's side, and that has all sorts of interesting implications for tracking evolution and our ancestry. So this means that only daughters will pass their mitochondrial DNA on to future generations. Another interesting fact about mitochondrial DNA is that there's an awful lot of it. In each of our cells we have just one set of nuclear DNA. We have our 46 chromosomes, but we have lots and lots of mitochondria in each of our cells, and in every mitochondria there's many copies of DNA. So we have lots and lots of copies of mitochondrial DNA, whereas we have only one copy, if you like, of our nuclear DNA. So it's this, as it says here, this huge abundance of mitochondrial DNA, as well as its small size, makes it excellent for use in forensic studies and also in archaeological studies. So if you can find ancient DNA samples, which are hundreds if not even thousands of years old in some cases, whereas the nuclear DNA may have degraded and be not useful, because there's so much mitochondrial DNA, it's possible that you may find some in good enough condition to test. In this slide we can see the inheritance pattern of mitochondrial DNA. So keep in mind, although males and females both carry their mother's mitochondrial DNA, only the females can pass it on to their children, since they're the ones contributing the egg. So what's the advantage for ancestral information about our past? Well, the interesting thing is that our nuclear DNA, as it gets passed on from generation to generation, gets mixed, because the nucleus from the sperm meets the nucleus from the egg to create a combined nuclear DNA in the zygote. Whereas, so it gets mixed up over generations. Whereas mitochondrial DNA remains unmixed, because there's a strict line of descent from mother to child only. So this means that our mitochondrial DNA is the same as our mother's and our mother's mitochondrial DNA for hundreds, if not thousands, of generations ago. So the mitochondrial DNA that we have in our cells is actually takes us back to the mitochondrial genetic code of our maternal ancestors from thousands of generations ago, and that's extraordinary. We're literally looking thousands of generations into the past when we look at the genetic sequence on our mitochondrial DNA. Let's have a quick look at the nature of mitochondrial DNA. So we should be familiar by now about nuclear DNA that has folded itself around histones and then looped itself and further folded itself into the structures we call chromosomes. So it tends to be long and linear in shape. Mitochondrial DNA, on the other hand, is much smaller and exists as a small circular loop of DNA, often referred to as a plasmid. These are also found in bacterial cells. So these small round circles of DNA are referred to as a plasmid. The other thing that makes it useful to study is that nuclear DNA is hugely long. So, as it says here, up to 247 million bases in length for nuclear DNA. That's a lot of DNA to study. Mitochondrial DNA, on the other hand, is only approximately 16,569 to 16,571 bases in length. So, much shorter and therefore more easily studied. The strongest theory for why our mitochondria and for that matter chloroplasts in plants both contain DNA relates to the idea that many years ago in evolution, you're talking perhaps hundreds of millions of years ago, that single-celled organisms, perhaps very simple original bacteria, the prokaryotes, may have formed a kind of a symbiotic relationship with larger cells. Alright, so this is called the endosymbiotic hypothesis. Endo meaning inside, symbiosis, as you know, means where two organisms live in very close relationship, each benefiting the other. So it could be that bacterial cells in the past lived in very close association, perhaps even inside larger cells, and provided some kind of advantage to those larger cells, and it could be that the larger cells provided some kind of food source to those early prokaryotes. And then, as this information suggests, it's possible that that over time this symbiotic relationship evolved into a state where the larger cells incorporated those bacteria into their very structures, and they became what we now today see as mitochondria. And because these bacteria carry these little tiny plasmids of DNA, that's why today we see these little small rings of DNA, mitochondrial DNA, inside the mitochondria as little circular plasmids. So, although it's only a theory, the evidence does support this idea of an early endosymbiotic relationship. The mitochondrial DNA contains the genetic code for just 37 genes. As it says here, 13 of the genes are responsible for producing proteins. 22 of the genes hold the genetic code to produce transfer RNA, and 2 genes hold the genetic code to produce ribosomal RNA. Hopefully you're reading that now and you understand what those things are. So the proteins, of course, are what will be made in protein synthesis. Transfer RNA will be needed in the mitochondria to carry amino acids to ribosomes to make the proteins, and of course the ribosomal RNA will contribute to the very structure of the ribosomes found inside the mitochondria. So the mitochondrial DNA is very important in the functioning of our mitochondria. Without it, our mitochondria would not be able to function in respiration. Okay, when we have a closer look at the mitochondrial DNA loop, there are 4 main regions, and they relate to what we just talked about a minute ago. The purple region here, which we'll talk about in a bit more detail in a minute, is the D loop, and all the other parts of the mitochondrial RNA code for what we just talked about. So the yellow regions code for proteins, which will of course be used in mitochondrial function. The white regions will code for transfer RNA, alright, these regions here, and the blue regions will code for the structural component of ribosomes, rRNA. Now let's finish the tutorial by looking at this D loop. The D loop, or what they call the hypervariable region of mitochondrial DNA, has a couple of very significant characteristics that contribute to some of our incredible genetic and evolutionary studies. The first thing to realise is that what we call the D loop does not contain any functional genes. So there's no coding in this part of the mitochondrial DNA for any functioning genes. So that's important to realise. When it comes to the structure of the D loop, the D stands for displacement. So D is displacement loop. And to put it fairly simply, that in the process of replicating the mitochondrial DNA molecule, a section of the double-stranded DNA molecule gets separated for a short stretch. So a section gets separated for a short stretch, and a third DNA strand that has a base sequence which is complementary to one of the main strands pairs with it. Alright, so we have double strand here, but we have the third strand of DNA which is separated from the original strand. So you end up with actually 3 strands of DNA in this loop here, this section. So you end up with triple-stranded DNA, which gives you this kind of capital D shape. So that's where the term comes from. To finish off the tutorial, it would be worth having a read through this section, and this will have implications when we talk about genetics and evolution a little bit later in the unit. Because there's no biological coding function in the D loop, it's a non-vital part of the mitochondrial DNA, and mutations, which are mistakes in the copying of the DNA, are important ways to track evolution over time. So as you accumulate mutations over time, this tells you something about how DNA evolves through history. The interesting thing about the mutations that accumulate on the mitochondrial DNA is, first of all, that any mutation that occurs in the D loop, because it doesn't affect any coding sections, never leads to death of the organism. It doesn't affect any critical function. So those mutations will tend to accumulate over time because they don't cause any harmful effects. They don't kill the organism. So any mutation that occurs in the D loop, in here, tends to stay in that section over long periods of time, and therefore gives you a very accurate kind of understanding of how mutations have accumulated, both good and bad possible mutations, over a long period of time through history. So have a little read of this section here, and this might make a little bit more sense as we talk about how mitochondrial DNA is used in genetic and evolutionary studies a little bit later in the course.